Now we will have the pleasure to hear Richard Furie. He is Chief of the Division of Rheumatology and Allergy Clinical Immunology of North Shore, New York, and Professor at STEM University and member of the ACR Board of Directors and Lupus Foundation of America. He will talk about failed trials, failed treatments, and the same. Thank you, Richard.
Thank you. Good morning. So I'm going to talk about lessons learned, which is a very polite way of saying we made a lot of mistakes over the last 20 years. Disclosures? But I'm going to put you to work even though it's 8 o'clock in the morning. So here's a question for you. So the Argentinian national football team has an international reputation in World Cup play, clearly many more wins than losses. So what has a losing record? Lupus clinical trials, we have 3 wins and lots of losses. Sorry, Mexico. These are the topics that I'd like to discuss. And we'll start out with ensuring clinical activity represents active lupus. Here's another question for you. You can redeem yourself. It's 8 points can cause more pain to sponsors than to patients. What is it? Okay, a lupus headache. It's worth 8 points on SLEDAI. So as you know, our extrarenal trials pretty much use SLEDAI or use BILAG and require a certain amount of disease activity. Let's go back in time to the phase 2 belimumab trial, and though this is a very busy table, I just want you to focus on the red. In this particular trial, at baseline, the mean SLEDAI score was 9.6. But notice that only 72% of the patients were serologically active. And in my book, if someone is clinically active, they should have autoantibodies. The end result of that study, as you know, is that neither co-primary endpoint was reached, but the post hoc analysis demonstrated clinical efficacy in the serologically active patients, which represented 72% of the cohort. So the rules for entry were changed not only for the belimumab phase 3 trials, but for all trials, and that is that seropositivity was required at screening. Let's jump to epertuzumab, anti-CD22, the phase 2b EMBLEM study. There were 227 patients in this particular study, and at baseline, 15 had lupus headache. That accounts for 7% of the cohort. So how does one prevent, I'll call it contamination of a cohort by subjective or highly weighted items? Well, one could require serologic confirmation as is being done. There could be an adjudication committee to hear all the cases. Photographs for skin disease, joint counts for arthritis. Eliminate some of those high weighted items such as the lupus headache, or require a PD marker such as the interferon signature. All right, let's move on to discuss sample size, effect size, and statistical significance. Here's another question for you mathematicians. So if x is 11% and y is 10%, when is y superior to x? And I thought I knew the answer, but David Isenberg actually supplied another answer about a year ago. He said at tax time, and he's correct, but also in the Bliss 76 and the Lunar trials. Here are the top-line data from Bliss 76 and Lunar, and you can see that the effect size, roughly around 10 to 11% in each of the 2 trials, is about the same. Yet one study, the Bliss 76 trial, was a success, and the Lunar trial was deemed a failure. And you can see the p-values. So what was the difference? In the last column, you'll see the numbers in each arm. So in Bliss 76, 275 subjects in each arm, and in Lunar, 72 in each of the 2 arms. So how did these numbers get determined? In the Bliss trials, post hoc analysis of the phase 2 seropositive subjects showed differences between placebo and treatment in the teens, regardless of which metric one looked at. So going forward to phase 3, the estimate was that one would need about 270 patients per group to detect a 14% difference in the SRI. Whereas in LUNAR, they were going for a home run. The estimates were that 30% of the placebo group would achieve a complete response. With the addition of rituximab to MMF, 50% would achieve a complete response. And you can see the numbers for the partial renal response. So based on these estimates, only 70 patients per arm were required. So in determining these numbers, be realistic. Try to reduce the sample size. The landscape right now is incredibly competitive. Good patients are hard to come by. For extrarenal trials, enrich for patients that can inform. Go after the patients with the high SLEDAI scores. We'll hear from Dr. Merrill about innovative trial designs, but it's crucial also to limit the number of treatment arms, so know the dose going into phase 3. Also know the placebo response. There's an initiative sponsored by the Lupus Foundation of America and headed by Ken Kalunian to collect, pool, and analyze placebo data from all of these clinical trials so that we can overcome some of these trial design challenges. Next to endpoints. Here's another question. When is a responder not a responder? In the Explorer and abatacept lupus nephritis trials. So you've seen some of this recently. These are data from the Explorer rituximab trial, and what it shows is that whether you got placebo or rituximab, patients got better. There was a 50% reduction in the composite BILAG total score. Yet, if you look at the far left in the circle, over 70% of the subjects were considered non-responders. Why is that? Well, that's on this slide. These are the definitions, and I'm not going to go into the details, of response, very rigorous and very complicated. Turning to the abatacept lupus nephritis trial, this is a schematic of the design. There were 3 arms, a high dose, a low dose, and placebo. Here are the top-line data. The time to confirm complete renal response was the primary endpoint, and you'll notice 2 things on this graph. Number 1 is that the curve superimposed, and number 2 is that the response rate was low, in the teens. And it gets even worse if one looks at the complete renal response confirmed at months 11 and 12 at the end of the study. And you see single digits, in fact low single digits, as far as response rates. This is all because of design issues. The endpoint was very rigorous and there were some mistakes made in the endpoint. Protein creatinine ratio of less than 0.26 was the requirement for response. The GFR had to be within 10% of baseline. And the mistake made was that if patients improved more than 10%, they were considered non-responders. There also had to be confirmation 1 month later. But modifying the endpoints, one sees different numbers. So looking just at week 52, at complete response, you see basically equivalence, but certainly higher numbers than what I showed you before. Correcting for that GFR of 10%, that is the patients who improve more than 10% being deemed non-responders, correcting for that, one sees a different set of numbers, 15 to 22% in the treatment group versus 12% in placebo. And using the LUNAR endpoint, one sees even better effect size. So the problem with clinical trial endpoints to date is that no 2 trial endpoints were the same. We've customized our endpoints. We're still wrestling with extrarenal trials. Should it be a singular endpoint or a composite endpoint? That is, should it be based on SLEDAI or BILAG alone, or should we use composite indices such as BICLA or SRI, or even a modified SRI with higher thresholds of response? So there's really no consensus for extrarenal studies. Certainly in lupus nephritis, where it's a little easier, the endpoints are converging on a very common endpoint, which is proteinuria based. So the attributes of an endpoint, they should be easily measured, valid, reliable, sensitive, clinically relevant, easily understood. Obviously, they need to discriminate treatment from a comparator, and standardization would be ideal, such as our rheumatoid arthritis colleagues have generated. Background therapies work. All right, here's a question for you. Has anybody gotten them all right yet? I may be old and cheap, but I am very effective for lupus and FDA approved. What am I? All right, steroids. Compound E, first used in rheumatoid arthritis in 1948. Back to the Explorer slide. There was a 50% improvement in the composite BILAG total score. Why was that? Because immunosuppressives had to be fixed during the entire study. It's because steroids were increased at the beginning of the trial and then tapered. We wrestle with steroids in our trial design. What should be the maximal dose at entry? What about during the study? Should they be tapered or should the dose be fixed? And if it's tapered, should it be a mandated taper or a voluntary taper? And if taper is mandated, should it be a strict or flexible schedule? What about flares? Should we allow steroids for flares? How long can they be given? How often can they be given? And the same kinds of questions can be asked about immunosuppressives. So what has been done? Each trial has handled steroids and immunosuppressives very differently. And the last point I want to touch on is regional safety differences. He was the ultimate hand washer, but unfortunately he was not a principal investigator in the Belong trial. Dr. Semmelweis, the ultimate hand washer. We've been confronted with regional safety issues. Data from the ALMS trial and from Belong pointed to Asia for some problems. There were more deaths in Asia than rest of world in the ALMS trial, and more opportunistic infections in the Belong trial. So along the way, we've made a lot of mistakes, we've learned a lot, and trials of new biologic therapies have taught us to be humble, very humbling, logical, and perseverant. So thank you, and I'll handle any questions now or at the end.
Thank you very much, Richard. May I put you a question? Is there any difference between phenotype and serologic markers of the physiopathologic process in the failed trials? Are there differences between the clinical endpoints and the serological? And should we move to look for more markers of the physiopathologic process?
Well, we've certainly learned from the belimumab trials that there's greater effect size in patients who are more active, so say Selena Slidi greater than 10, and also patients, and that could be the low complement, high DNA antibody patients, sort of a surrogate for those patients with high Selena Slidi scores. So I think everybody is chasing those high disease activity patients for their trials.
Dan Albert, Dartmouth. Thanks, Rich. That was excellent. I want to address one additional problem that's sort of imposed on us by the FDA, and that is that we have a single primary outcome and that it's a landmark study. It has to be done at a certain point in time. So a dichotomous outcome at a specific point in time is a very statistically inefficient way of looking at a very complex disease, and a much more broad approach would be something like area under the curve of a composite index. And if you reanalyze many trials with that kind of outcome measure, they become statistically significant.
I missed that. Was that a question or a comment?
That's
I appreciate
a comment.
the comment. Okay. I can't see.
I think we're done, Rich.
Okay,
Thank you very much indeed. Thank you.